can start our webinar. So my name is Wolfgang Miesbach from the University Hospital in Frankfurt, and I welcome you to this webinar. This is a joint webinar from EAHAD together with EHC. And the title of this webinar today is Hub and Spoke Model and Practical Experiences from Europe. So this is the agenda of the meeting. Today we have 3 presentations from 3 very distinguished speakers and also experienced hemophilia treaters who also are experienced in new treatment options and gene therapy. The first presentation will be given by Bulent Zulfikar. Professor Zulfikar is hemophilia treater of the University Hospital in Istanbul, and he recently has published a very interesting article about gene therapy and hemophilia literature review and regional perspectives for Turkey. And after his presentation, we learn about experiences from France. And this presentation will be given by Professor Yesim Dargaud. And Professor Yesim Dargaud is professor in the Clinical Hemostasis Unit and Haemophilia Comprehensive Care Center at Lyon University Hospitals in France. And the final presentation will be given by Professor Peter Kampmann. And Peter Kampmann will tell us about the experiences he made with gene therapy in Denmark. Professor Kampmann is consultant specialist and head of the leukemia unit at the Copenhagen University Hospital. And after these 3 perspectives from 3 different countries in Europe, hopefully we get some more impact about the hub and spoke model, how it works, and how different aspects of the hub and spoke model will become more important in the future. There would be a panel discussion, and this panel discussion would be moderated by 2 persons who have invented the term hub and spoke in terms of gene therapy. So this has been first mentioned now some years ago by Flora Peyvandi and Declan Noone, that gene therapy could be carried out in the most efficacious and most safe way using a modifiable hub and spoke model. And after this panel discussion, hopefully we will have some more time to answer some questions coming from the auditorium. Now, very shortly, I will give an overview about the hub and spoke model. Here are my conflicts of interests. And you know, in hemophilia, we have in Europe, we have about more than 400 hemophilia centers. And these hemophilia centers present a great variability. So the variability is according to the number of patients per center. So there are some centers with more than 300 patients with severe hemophilia, and there are also some patients with only less than 5 patients per center. The centers also differ regarding the facilities. So in the laboratory field, there are centers with a 24 hours access to inhibitor testing, for example, and also using different reagents for the different correlation measurements. And there are also some centers with poor access to laboratory. It's the same for on-call care. There are some centers with 24 hours call, and this is not always possible, of course. There are also some differences regarding centers per country. In some countries, there's only 1 center, and in other countries, there are more centers. And this is because there is no generally accepted definition of a hemophilia center. There are some different names like Haemophilia Center, or Comprehensive Center, or Reference Center. And to facilitate this problem, and also to enable some classification criteria and certification process, EAHAD has started very well a certification process, and according to this, there are now more than 100 comprehensive care centers fulfilling certain classification criteria at the highest level. And in Europe, there are also more than 40 European hemophilia treatment centers also fulfilling certain classification criteria. This is mostly then in the collaboration with comprehensive care centers in Europe. Now, when we speak about gene therapy of hemophilia in clinical trials, there are several preconditions which on the first view are quite unusual to hemophilia centers, but are very necessary to be carried out because of the innovative aspect of gene therapy. First, the site should be selected by the sponsors, and it would be a control function by the regulators. The reimbursement is done by the sponsor, and there's also a standardized patient selection. Also, the informed consent is done on a standardized way. The very much important and intense follow-up period is standardized, including laboratory investigation and how safety problems have been managed. Finally, the data would be collected and also managed together with the monitors, and the whole process there would be done a supervision by the sponsors. Now, after the first approval of the gene therapy drug for hemophilia in Europe, we are faced with new challenges for our hemophilia center when we think about treat our patients with gene therapy. Unlike other treatment options for gene therapy, an extensive information education of the patient, also the team members of the hemophilia centers, should be done together with an extensive education, and this with the involvement probably of experienced psychologists. There should also be done some specific eligibility tests, not only regarding laboratory levels, so in terms of AAV, anti-AAV antibodies, but also some other tests, and finally concluding with an informed consent from the patient. The administration of the gene therapy construct is not really the most complicated issue, but there are some challenges, as well as possible infusion-related reactions. They can occur, and in most cases they can be managed quite well. Then after the treatment, variability of factor expression is expected, and this should be monitored quite closely to decide when to stop prophylactic factor treatment. After the treatment, some short and medium and long-term adverse events can occur, and for most of this, a close cooperation with experienced hepatologists and immunologists would be necessary. Finally, long-term cooperation with patient in the follow-up period is key, and for this degree of compliance and adherence is necessary. Data management should be done for long-term after gene therapy, and this is the most appropriate way to collect all possible adverse events, even if they occur long-term after gene therapy. And finally, direct and indirect reimbursement for the administration of gene therapy and follow-ups is still a very complicated issue. We see this now actually in the discussion we have in Germany. I'm very sorry, I'm a little bit, okay, I will come back now. Yes. And throughout the clinical trials, it became clear that there would be different responsibilities of different centers according to the knowledge and the facilities. So in clinical trials, there were some dosing centers and there were some home or follow-up centers. And in some kinds, there was also the possibility to contact supervisory or coordinating centers. And this was very useful, especially if centers were only able to complete one or any combination of two tasks. It was possible to partner with another center. In this scenario, the necessary work needed for gene therapy could be completed. So there are some different roles of hemophilia center in gene therapy. And different questions should be answered before. Which centers are responsible for the patient at each stage? How are dosing centers defined? Who is responsible for follow-up care and guidance? And should the patient be transferred between centers during follow-up? How are decisions relating to laboratory results and adverse events made? And even within clinical trials, there was no one single answer to this question. So this was managed by the interaction on an individual basis between the centers and related to the individual patient. And this is a paper about the hub-and-spoke model, which has been proposed by Flora Peyvandi and Declan Noone. And this is a joint publication between EAHAD and EAHC. And in this paper, once more, the challenges of gene therapy for hemophilia center have been documented, as well the responsibilities of the hub-and-spoke center. And the national hub center have been defined to prescribe and manage gene therapy, and this is done exclusively by expert hemophilia comprehensive care centers, whereas spoke centers has been defined as local centers with the responsibility to do all the pre-treatment activities, but also the follow-up by monitoring. This can be done in close communication by the spoke center with the primary expert hub center. So the interaction and communication is very much important because of the need for regular laboratory measurements before gene therapy, but also after gene therapy. The travel of the patient has to be managed between the spoke center and the hub center. And there should also be some defined criteria for communication, cooperation, how to manage and deal with safety alerts, for example, when to stop regular factor replacement, what level of ALT elevation would trigger immunosuppressive treatment. This is really not an easy question, and when to taper or escalate immunosuppressive treatment. So using a hub-and-spoke model with well-defined responsibilities of the different centers would enable the centers to offer an optimal level of treatment using the knowledge of the hub center and to transfer the knowledge to the spoke centers with the possibility to monitor and care of the patient in his local area with a continuous support and counseling by the hub center. There should be an agreement on the distribution of the different talks regarding who is responsible for the laboratory monitoring, for carrying out the immunological tests, anti-AAV antibodies, and who is responsible for data collection and entering into the registries. And not only the role of the hemophilia centers need to be adapted with gene therapy, but also the role of each member of the multidisciplinary team. So the hematologist is responsible to check the outcome and also to initiate the immunosuppressive treatment. The nurse is very much important to manage the expectation of the patient, to support the shared decision-making, but also to coordinate the cooperation, the communication between the center and the patient, and also between the different centers caring for the patient. The psychologist may be more important than in earlier times to assess the dedication and compliance of the patient, also to assess some psychological levels baseline. The physiotherapist is also very much important to assess the risk of physical activity and musculoskeletal assessment. The hepatologist is probably the most important member we have now in our team to monitor the liver health before gene therapy and also after gene therapy. And we also need the immunologist to better understand the immunological reaction and liver reaction after gene therapy. And with this short introduction, can maybe start with a presentation from France.
Good afternoon to everyone, and thank you for the kind invitation and the opportunity to share our experience in France. As you can see on this first slide with the comments of friends and colleagues from other European countries, but also with the question and remarks from my friends in France, you can easily realize that to implement an hub and spoke model in France was not an easy battle won in advance. To my eyes, the hub and spoke model is first of all about sharing of experience and learning together. Some of us, we have had this clinical experience of gene therapy and the clinical experience of handling with the liver toxicity of AAVs during the phase 2, phase 3 studies. And hub centers should have this clinical experience of gene therapy, and they should have the capacity with their multidisciplinary teams, institutional approvals, appropriate laboratory facilities, and will to receive patients of other centers and share their experience. In France, we have slightly modified the original concept of hub and spoke model is originally defined by EAHAD and EHC and adapted it to our national organization and infrastructures. As a result, we gave more responsibilities to the spoke centers. In addition to the role of spoke centers for patient selection, pre-screening, eligibility assessment, after gene therapy drug administration, spoke centers will be also at the first line for patients' follow-up. But for doing this, we generated national SOPs, standard operating procedures, where we have clearly defined what will be the clinical care pathway for gene therapy in hemophilia, and how will be the structured follow-up for the patients. All this educational material for health professionals will be published at the beginning of the next year in French, in the in the Journal of the French Society of Hematology and will be shared with all hemophilia centers in France. Of course, the corresponding hub center will be actively involved in the follow-up of patients at the site of the spoke center and provide guidance, in particular in patients who will have, unfortunately, side effects such as liver toxicity and who may need immunosuppressive therapies. And in addition to these two levels of security, the home site follow-up and the hub center guidance, we also set up the possibility to discuss difficult cases at the national multidisciplinary case conferences organized by the French Reference Center of Hemophilia with a multidisciplinary experienced panel of experts, including hematologists with experience in gene therapy, but also experienced hepatologists with gene therapy and immunologists. For information of the patients in spoke centers, we have closely worked physicians and patient organizations to generate firstly a national survey for French patients to determine their expectations from gene therapy and also to determine their level of knowledge on gene therapy. And in parallel, we are currently working on educational material for patients, which will be used in all French hemophilia centers to provide appropriate information on gene therapy to all patients. And the key messages from this educational material, common material, will be extracted as a flyer, which will be given to each patient at the end of the first information visit. And patient can continue to use this material and share it with his family, his friends during his decision-making period. If the patient is motivated and corresponds to the eligibility criteria, we think that the eligibility assessment should be performed very rapidly with anti-AAV neutralizing antibody testing. But after that, each patient should have sufficient time to make their decision, because this will be a decision of their life for a treatment which is able to transform their disease. And if this reflection period takes longer than 3 months, then we decided to repeat the anti-AAV neutralizing antibody testing. For hub centers, the administration of the gene therapy drug is relatively simple. Their real role will probably be to provide guidance, share their experience during the follow-up period with spoke centers, and in particular for patients who will have side effects and liver toxicity. From this perspective, it is mandatory to have informed and experienced hepatologists in our multidisciplinary team. Another challenge for hub centers will be to try to establish a relation of trust with patients coming from other centers that we don't know. So we decided to have a systematic pre-gene therapy visit at the site of the hub center, where we will offer to patients to know the medical staff of the hub center, to answer their questions, complete information if necessary, talk about the informed consent, and go through the eligibility assessments. Concretely, we have 36 hemophilia treatment centers in France, and at least 5 of us, we have clinical experience from clinical trials of gene therapy, and these will be the 5 hub centers for all country. The day of the gene therapy, patients will be received early in the morning, and after drug administration, we plan to keep them under medical observation during 6 hours for any acute immediate complication of the drug administration, such as fever or anaphylactic reactions, hypersensitivity, headache, of any other complications. If in the absence of side effect, patients will leave the hub centers the same day in the afternoon. In hemophilia centers, we are used to cooperate and work with colleagues from hepatology department because of the hepatitis B and C in our population. But for gene therapy, the role of hepatologists will be key, and they will be involved in the process during the entire course of gene therapy. They will help us to assess in a personal way the liver status of our patients before gene therapy, and they will also help us to manage the liver toxicity after gene therapy. And we have also included experienced, informed hepatologists on AAV gene therapy in our national panels for generating guidelines, but also for the multidisciplinary case conferences to discuss different cases. And with regard to the data collection, we have decided to use our national database on inherited bleeding disorders, France-COAG. All hemophilia centers, they are used to report data of their patients on the rhythm of the usual visits. For gene therapy, we will collect specific data, and we decided to collect lifelong, every 6 months data on these patients, and more frequently during the first year. This is schematically the plan that we have to implement hub and spoke model in France. We decided to give more responsibilities to the spoke centers because we don't want to lose patients during the first year of follow-up, where the follow-up is very demanding, and we don't want to make them travel several times between the hub centers and spoke centers. But the real uncertainty for us now is the approval and also the reimbursement of the drug. Because in France, the approval of the drug does not automatically conduct to its reimbursement. And we have another independent expert committee, national committee, which decided the SMR level. What does SMR? SMR evaluates the added value of a new drug compared to the existing therapeutical molecules. In practice, it analyzes what the new drug really improves for the patient care, and what are the safety issues with the new drug. What is the severity of the disease that we are talking about for hemophilia? And are there any alternative treatments or not? And taking into account all these parameters, this committee will define a level of SMR going from major therapeutic progress, level 1, to important, moderate, minor, or no therapeutic progress. To give you a few examples, emicizumab had an SMR level 2 because of the significant reductions of ABR, subcutaneous administration, the possibility to use in patients with and without inhibitors. But EHL factor 9 molecules had an SMR level 4, minor therapeutic progress, because they consider that this is just the improvement of quality of life with less frequent injections. And EHL factor 8 molecules had an SMR at 5, so no therapeutic progress. And this SMR level directly has an impact on the price of the molecule and the reimbursement in France. Of course, all these SMR records are top secret, and we have no information yet on the reimbursement of gene therapy in France. But from the medical perspective, to implement gene therapy in France, what we definitely need informed, experienced hepatologists with us that we should include in our multidisciplinary teams. I'm not sure that we need central labs, but hematologists should be really aware of the between center variability of the factor dosages, factor testing. And we are thinking that the laboratory monitoring should be always done in the same lab and with the same method. And even with the chromogenic assay in the last ECAT report, there was more than 18% of variability for factor VIII levels, around 20%, and it was more than 30% of variability between centers for low values of factor VIII, less than 5%. So this can be an issue and a source of worry for patients. And we need patients' adhesion for this very demanding follow-up for during the first year, and we need a close communication and cooperation between centers. And we should learn more about with a lifelong data collection. And to conclude, I think that this care model should be dynamic and should be subsequently adapted to the new knowledge that we will all generate together on the safety and all the efficacy of this new therapeutical modality. Thank you very much.
Thank you very much, Professor Dargaud, for this very nice and interesting presentation. I think a lot of possibilities for discussion afterwards. And the next presentation will be given by Professor Peter Kampmann about his experiences in Denmark. Peter, please.
Hi, thank you very much, and also for the opportunity to share our experience from Copenhagen. I just need to have the control board of my conflicts of interest, also regarding gene therapy. And as we leave those on, I just say thank you to Professor Dargaud. I love your first slide on hub and spoke is and is not, because it highlights sharing and safety, which I think really is the key to what we're talking about here. I'm here to talk about the Danish gene therapy experience also in the hub spoke model. As you will see, I represent sort of a spoke center, but still having experience with 4 patients. The facts covering my center is that it's situated in Copenhagen, and we cover a population of 2.6 million citizens, which brings us to a point where we have approximately 450 persons with hemophilia or combined von Willebrand defects and platelet defects. So in comparing to other centers, you could say that the other Danish center, which is in Aarhus, is approximately the same size. And at this point, we have experience from 4 cases of hemophilia B gene therapy since 2014, 1 being a phase 1/2 protocol and 3 of them being the latest phase 3 protocol. I have not done any gene therapy infusion on my side, so all cases have actually been infused abroad. First in 2015 in Frankfurt, who's at Professor Wolfgang Miesbach, and then in 2019 in the Netherlands with Professor Michiel Coppens. So in that respect, I represent one of the spoke centers that you would talk about in this model. All of my cases have been in clinical controlled trials, and you probably know from the blood publication that the phase 1 is published, and right now we are in the writing phase of the phase 3 trial. Actually, what I would propose to you now is what I think that I have experience with, and you would see that infusion is, of course, the least part of it, but the very long process of having your patient into gene therapy is, of course, about information, screening, inclusion, and the pretty long lead-in that you would see in a clinically controlled trial, where patients are, as a matter of fact, their own controls, having a rather long lead-in, and also that in the protocol setting, a lot of time to talk and think things over. We have talked about the lead-in of the treatment once it gets probably approved, and we would also expect to have a pretty long timeline for repeated questions and thoughts before patients get fully cleared to go into gene therapy. So in that perspective, the protocol setting could look a bit like real life afterwards. We've done a lot of preparations for infusion and travel, because this will actually be one of the new challenges to a spoke center. Of course, there's been sort of a last checkup before my patients went abroad. We had to do a line of rescreening, but the transition to another center itself poses a challenge, specifically if you choose to send your patient abroad to another country. You could probably think about insurance. What about backup? Should they be followed by a spouse or a friend? There's a lot of practicalities to it, and it's sort of another aspect which the hub center will of course have to deal with as the receiver of the patient. My infusion sites were in Frankfurt am Main and in Amsterdam, and as I tried to write here, my patients would arrive on a Tuesday, being dosed on a Wednesday, and then returning to Copenhagen on a Thursday. So throughout the whole setting of the protocol, having a lead-in of more than 6 months and the screening period before that, and the follow-up of 3 to 5, 10 years, the infusion itself is only a matter of a travel of 72 hours. Still, it means that to the patient, those 72 hours are crucial, and it really makes a great impression in their minds once they go back. Once returned, I would be the one to do the early assessment of effect, follow side effects and hepatotoxicity, and doing the long-term follow-up. And in that aspect, if you think of the hub-spoke model after approval, you would of course think that I would have to correspond with a hub, whereas in this case of the clinical trial, my hub expertise was of course the medical monitors of the sponsor. So we've talked about this in the Nordic settings, because could we call this hub spoke if you look at Scandinavia or the Nordics, or have we debated whether we should be called infusion centers? If you look at the organization in the Nordic countries, we're based on very few and very large centers, none of us being sort of smaller than the others, and none of us sort of lacking expertise. And all centers in principles could be hubs. The question is, who will infuse? And we have debated whether this should be seen as sort of a national or a regional aspect, because if you look at the geography of the Nordic countries, you would find that in specific, the 2 Danish centers combined with Lund-Malmö in south could be considered one region, maybe also including Gothenburg, whereas you go further north, you'll see the capitals and the centers a bit more dispersed. And in my situation here, if I was to send patients to other centers, they would go for 1 hour's drive to Lund-Malmö in Sweden, whereas if they should go to Aarhus, that would be 3 hours in a car. So we have been debating whether you could also call this regional. The other thing is, all our centers could be considered comprehensive care centers, and to some doctors it could feel a bit weary to be designated as a spoke if your neighboring centers was maybe smaller or had less members employed. So that's really why I like that first slide of Professor Dargaud, since it actually is not a matter of is or is not. This is a matter of safety and collaboration. So in my mind, I wouldn't mind being the spoke also in the future, even though my colleagues in my neighboring centers would be infusion centers or hubs in this type of nomenclature. The big challenge probably being legislation and reimbursement. That could call for national hubs, and that's probably where we'd see some of the challenges in the Nordics, even though our centers and our culture and languages are pretty much the same to others. We would still have boundaries, and we will still have legislation to maybe be an obstacle, so that we would in practice have to be forced to have at least a national hub or national infusion center, but time will show. So this was the doctor's perspective. If you talk to the patient here, what do they experience and what is important? As you all know, first of all, relations. I'd like to have the relation with my center, my life, and I'd like to have my gene therapy follow-up at least close to where I live. If I go to a hub or infusion center, what I want is smooth infusion and, of course, professional and experienced staff. And this is, in my opinion, what really speaks in favor for having hub-spoke also in Denmark, where we have two centers of same size, because experience and number of infusions do matter. On the other hand, I should say thank you to Wolfgang Miesbach and Michel Coppens, because my patients have been very satisfied and felt very warmly welcomed at the centers, even though at that point, even as I was, we were all beginners, but they had a very professional experience. So that could, for all of you others, probably count to the fact that because we are also dedicated, I think that patients will find infusion smooth, professional, even though you're not experienced. So all aspects of gene therapy, except infusion, should actually be undertaken at the patient's comprehensive center. In that perspective, I think we could do all the steps except from the infusion, and that would support the fact that the patient should be followed up and seen largely in their own center. But the infusion, in my mind, should be undertaken at a site with specific experience. That brings me to the fact, what is a hub in a Nordic perspective? And I think this is very much in accordance with what we have seen in the last presentation, because there is a fact or a problem regarding GMO permissions. It is rather burdensome to get those permissions. And I would say that in Denmark, if one of our centers should get that one, I would happily send my patients to the other center just to have only one place where we should go through that process. There has to be specific pharmacy expertise on handling gene therapy. And of course, that goes as well with the communication with the medical company providing the gene therapy product. But we have mentioned the hepatologist, and that's where we may have a special word for hubs, because the number of patients will be scarce, and the expertise will, for the hepatologist, will take some time to gather. And even in larger countries than Denmark, or if you consider the whole Nordic region, we would probably get localized expertise on these specific low-grade inflammation problems, which we could see probably or maybe in gene therapy patients. So far, we have not seen many complications and side effects, and the treatment has actually shown itself from its best side. But over time, as numbers go up, we probably should see that there could be a need for having experience of the more subtle and rarer types of complication that we would see. And in that case, a hub would probably best serve as the knowledge base for those complications, and also being the center for the regional database and the collection from other spokes. And looking internationally, we have to have representatives in a national network. So that was my short talk on that. I think it underscores the details of my predecessors in the talk. On the other hand, I've not been a hub, and I feel fully content about the product I've made so far.
While I would like to thank my owner for giving me the opportunity to take part in your meeting with my knowledge and experience, I would also like to congratulate Mr. Declan Noone and the organizing committee for raising an important issue. The use of factor concentrate in the treatment of hemophilia is still a cornerstone, but they also carry the risk of inhibitors. Subcutaneous molecules that have been used and waiting to be used in the last 5 years are changing the concept. Standard and modern approach have shown efficacy in reducing the rate of bleeding, but their usage may be limited by the risk of thrombogenicity, and both still require lifelong injection without restoring normal hemostasis. However, there are also possible some undesirable side effects. More than 400 centers in Europe provide comprehensive care for hemophiliacs with multidisciplinary team. clinical trials are currently underway on the gene therapy in hemophilia A and hemophilia B, more than 10 countries. Although the study were started by academic institutions, they were quickly spread by the pharmaceutical industry. It continues to be implemented in 7+5 countries, 12 countries, and 31 centers, especially those supported by BioMarin and Pfizer. Very recently, European Commission has licensed Roctavian, manufactured by the BioMarin, for use in severe type adult hemophilia A. Thus, the discussion on the principle of intensive use, the problem of registry and follow-up in terms of safety and efficacy in the near, middle, long term have increased. The hub and spoke model is one of the most efficient and applicable approach to the subject. This is the Turkish data. There are 6,000 hemophiliacs in Turkey with 2015 figures. Today, we know there are more than 5,000 hemophilia A and more than 1,000 hemophilia B. Over 50 clinics provide hemophilia care. 9 of them are certified by EAHAD, EAHC. Gene therapy is applied in 4 cities. Currently, 20 patients are followed in clinical trial more than 15 months in Turkey. 13 of them are the hemophilia A and 7 of them are hemophilia B. Most of them are followed in Egen University by Professor Kavakle. Studies continued at the centers in Adana, Gaziantep, and Istanbul. In Egen University, the pediatric hematologist Kavakle and Dr. Shine, adult hematologist, and they are in hemophilia A, and they have study related to hemophilia A and hemophilia B gene therapy. The provided factor 9 level is between 15 to 30%, and the factors 8 level is around 50%. This year, we published a review on gene therapy by all centers together. We examined the history of gene therapy, the latest situation in the world, and of course, the expectation. We also discussed our country's experience and the approach of our patients, families, and health system. In this section, I will talk about the difficulties experienced as a country. The first line of this is that the presence of antibody against the AAV virus. AAV virus was found to be quite high in our patients. In Ferla's study, you have seen these slides, this rate was 14 in Italy, 27 in Netherlands, and very high, 67% in Turkey. This is my center experience. We are involved in both phase 2 and phase 3 studies. As you see, I screened 14 patients before clinical study. Unfortunately, only 1 had no antibodies. For this reason, we lost the chance to include more patients in most of clinical trials. The second important problem is the presence of hepatitis B and hepatitis C. The incidence of viral hepatitis is high. The rate is over 20% in children and over 60% in adults. It is pleasing that the rate of carrier has decreased with vaccination in the last 25 years. There are no HIV positive among them. However, active viral hepatitis or chronic hepatitis is exclusion criteria in gene therapy. Although there are multidisciplinary teams which are very important for gene therapy, collaboration and engagement of scientists, manufacturers, governmental regulators, health technology assessors, patients and families, national advocacy groups is also crucial. Unfortunately, the information about the gene therapy, especially healthcare professionals, is not at the desired level. While the rate of this who could not trust their knowledge about gene therapy was 44% in Dr. Glenn's study, and also 40% in Dr. Flora's studies. Finally, I would like to share a few features about the situation of my citizens. Hemophiliacs living in Turkey have excessive expectation and they have been dissatisfied with and not applying the existing. And also our personality structure that expects to be touched, given excessive attention and share responsibility. And also their protective approach, they have a protective approach of their families, their mothers, especially their mothers, keeping the hope of getting rid of the disease and its complication one day. Of course, we are glad they are full of hope. It's good. The clinical and psychosocial differences of the people should also taken. We know that the rate of bleeding and rate of viral hepatitis, rate of childhood viral diseases, and also hepatocytosis are high in my country. As a conclusion, gene therapy is a potential life-changing and possible milestone in hemophilia. With the huge efforts for overcoming the current obstacles, it can be expected that patients will significantly benefit from the gene therapy with remarkable increase in their quality of life. Continuous training on the subject and cooperation between the stakeholders should always be maintained. Also, it should never be forgotten that there are cultural differences between the people, between the country. And I would like to be among you. I will be on the plane to attend the Hematology Congress in Baku right now. I will be at 19th International Haemophilia Congress of Turkey, of which I am the co-president on November 18. Mr. Mahoney, Brian Mahoney, will attend our Congress with video recording and share his experience on gene therapy. Thank you for your kindness to listen to my video recording. We will discuss the gene therapy at the 19th Haemophilia Congress of Turkey. Thank you again.
Thank you, Bulent. It was very good to be able to hear you and to have seen also the amount of the work actually you have done in Turkey is amazing, and is really important, the data you've shown in terms of the positivity of anti-vector antibodies. And that is bringing me to one of the questions on when we are talking and counseling with our patient and explaining what is gene therapy. So between the 2-3 steps of consultation and decision of the patient, there is also the step that you have to make a test and to see if the patient has the antibody against the vector or not. And it's a long process. And if a person with hemophilia is just thinking and at the end deciding to go for the gene therapy, and then you come up and to say, I'm sorry, the antibody was positive. And this procedure really, I think, to be taught very well. And there was a question from Lutz talking about how you think, I think it was Lutz, how we have to think about this psychological aspect and what should be the role of the psychologist. And Yesim, do you want to tell us how you manage the stuff regarding the psychology aspect in France? And maybe Peter, you want to tell us in Denmark eventually in future.
Well, thank you. We are discussing on this point. There are at least two perspectives, I think, in the question. The first one is, again, this is a decision of life, and patients' expectations are very important. And really, the gene therapy can modify their disease. So I think a discussion with a center, in the Haemophilia Center of Psychologists or other psychologists outside of the center, but for him, we have communications. It would be really should be offered to the patients. And from the medical perspective, in case of need for corticosteroid therapy, for some patients who may have some psychological fragilities, it would be also a need to evaluate if a corticosteroid treatment can decompensate this situation or not before telling them we can go or not. But it is not a contraindication, I think. And as the active hepatitis B or C, gene therapy is not an emergency situation. We can offer patients first a therapy before and then discuss again gene therapy once their psychological problem or their hepatitis is controlled.
Thank you. Peter, do you want to add something in the Nordic area, why you guys are, how you are thinking to manage that?
Yeah, first of all, I'd say that I agree that we should very early on case find out if the person is fully eligible from at least the objective criteria. And in my perspective, opening the opportunity to gene therapy for a single patient or an individual, we'll start out with running through the patient's history and at a very early stage doing all the testing that would probably exclude them, or at least having that exclusion criteria ruled out before going into the official opening and the very deep information. I think it does pose a problem that you lure the patients too far into gene therapy and then in the end finding out that there would be an exclusion criteria. We've not experienced that. I have had no exclusions at the early stage, but I used a lot of time trying to find out. And luckily, the trials that I was part of actually accepted low levels of anti-AAV antibody, especially in the phase 3 from what we learned in phase 1. And that made it a bit easier. And from that reason, I did not have to exclude anyone. What we experienced during the course of treatment was that once the patients have had their gene therapy, they had a literal confusion of identity. So what other people would say that it was sort of positive sorrow, or what you could say, because of getting rid of factor infusion still changed their lives so much that they had to rearrange themselves and the picture they had them on their own impression. And my patients went through that phase very differently. And looking at the next phase we're going into, I think I can handle the first expectation phase and the run-in as a doctor, and just by posing myself open and available for them. But the later phase, 4 to 12 months after infusion, that's probably where we should think what a psychologist could do for them. At least I feel like I could put myself available as their connection, their relation, and their doctor, but there may be perspectives that I cannot foresee, which they have to debate with the psychologist instead, just to rearrange themselves and get more safely through that pretty difficult conflict of their self-comprehension.
So I think that is actually really important. So my takeaway, there's 4 key phases. There's the eligibility and the psychological element that brings. There's the assessment after gene therapy, there's the assessment of steroids, and then there's that readjustment period of what the new life is going to be. There's another question here, and it's one that came up at our New Tech conference this weekend, which was, if I'm going to my local center and my infusion or my hub center is somewhere else, and they use different lab assays, how will that work? Because I'll go into my hub and maybe that'll be managed at one stage, and I'm told my level is 40. and then I go for my other assessment, and it's maybe 24, and you're using different assays. How do you manage those conversations with patients to help them know their factor level and what these mean with injection after gene therapy? Peter, do you want to take that one first?
I've had that conversation with all of them, and actually what I did was at my site to do both the one-stage APTT measurement, but also the chromogenic. And with at least the Padua variant of factor 9, you would see that there is quite a large difference in between. So we tried to consolidate our knowledge by doing rotem tech as well, and that was very explicable to the patient to see how the full blood coagulation analysis at least showed them that there was a difference, even though that was not part of the exploratory endpoint in the trials. In the end, what really made them sure about the gene therapy was their everyday bruising and small accidents, because no matter how you're treated and what you measure, in the end, what you gain confidence in is what happens to your body on the everyday basis. And that's sort of the other aspect of psychology here. It takes 3 to 6 months before they gain proper trust in their new treatment. And that doesn't matter whether it's 20% or 45%, whether the TEG is normal or subnormal. They have to experience normal day bruising and probably have an operation or two just to make sure it actually works.
Yesim, you wanna?
Yeah, we know that between one stage and chromogenic assay, there's approximately 1.5-fold difference. And if we change method from one determination to another one, it's expected that results will be different. So this is something that we have to discuss with patients and try to keep always the same lab with the same method during all the follow-up. I think this is an information that we have to give to the patients. And the result which correlates best with the clinical answer, I think it's the best one.
I think Wolfgang wants to say something.
Thank you very much, Flora. So I only wanted to mention there is not only a known variability of coagulation factor levels, but also of ALT measurement. And if you speak about continuation of measurement and sending the blood to the same laboratory, we also should consider that this should be done for ALT measurements. And regarding ALT, there's also circadian rhythm. So it makes a difference if the ALT level would be measured in the morning or in the afternoon, and this also should be taken into mind.
Since you are on talking, Wolfgang, there is another question regarding the registry. So do you guys think that your countries in the future will make the national registry, or there would be a link or duplication of the registry? Do we use the World Federation of Hemophilia Register? What's your thought about that? Wolfgang, I start with Germany, since you guys have to face up with this problem earlier than others.
Yes, thank you. So I think the WFH gene therapy registry is the most comprehensive and most complete, and also the most advanced registry. So this is done by the WFH together with scientific organizations, and it covers a lot of topics. So patient-related topics, gene therapy-related topics, all the possible adverse events. And this is a potential lifelong registry. So it's very elegant to include the data. And of course, in Germany also we have a national registry, and there is still ongoing discussion how the data from the national registry can be transferred to the global WFH registry. But I think there is no way not to include the data into the WFH registry, and the WFH registry offers the data inclusion of all the patient after approval, but also the patient within clinical studies. There should the data be included into this global registry.
And France and Denmark?
Well, I think it's really important to have, of course, national data, but we will all have very few patients who will have gene therapy, at least at the beginning. And it's very important to pool the data and connect our national databases with the WFH database in order to generate larger information and to learn more and rapidly on the efficacy and safety issues.
Thank you.
Yeah, I think this is going to be a really interesting legislation time for all of us around data protection, not just from a sharing data that helps us all improve, but also the contracts, the international collaboration, who pays for what, how does it work? So I'm going to combine two questions here, which is, what experience do we have from the organization of gene therapy in other rare diseases, and is it sensible to have a few hubs within Europe? And I think what we've seen with Zolgensma, which is the SMA gene therapy, there's about 18 centers in Germany, and then 4 and 6 in UK and France, respectively. That is in comparison to Zynteglo and Libmeldy, which has 1 center in Milan, I think, and then the others, Libmeldy was with the 5 centers across Europe. Do you think that type of approach would work in hemophilia gene therapy, or do you think that every country can possibly have their own national approach? I think it kind of follows on from Peter's point and his international collaboration. Do you think it's possible for the smaller countries to have hubs or infusion centers? Wolfgang, do you want to take that one first?
This is a great question, and it's really depending on several local factors, and especially on the reimbursement. So during clinical trials, there is no question how it would be reimbursed because it's sponsored. But after approval, I think it can become quite complicated if one patient from one country will be treated in another country. So, and you know how it works in Europe. So I'm a little bit skeptical about this question. Also, I think there could be some benefit.
I would say from a small center's perspective that that's why I put up the regional part, because if you look at the size of my country, you'd find that maybe in the end, in one type of gene therapy, we would have maybe 10 or 20 patients before the next one is on, and you would change provider. And then in the end, you would probably like one regional to have different medical companies as your associates, so that in an example, you could do one type in one country and the other type in the other. I think what you need is to have a center in your vicinity. No matter if the countries are small or if you have local reimbursement considerations in countries where different counties could have different legislations. I think it's just a matter of collaboration, but it will take time to put it on stage.
Yeah, I fully agree on this. We're getting very close to time. There's a ton of questions that keep coming in. How about let's talk about the where's my mouse? About the legislation around reimbursement. So there is a question on that, and as the health technology person on the I'll take advantage of my position. What we have in Europe is we have the S2 pathway and we have the cross-border directive, and we have seen that process work in clinical trials where patients have gone to other countries to get care, but in reality it gets quite difficult either through either pathway to get that legislation, because effectively what needs to happen on a local level is the product needs to go through a HTA, it needs to have a process on the delivery of care, and then a decision on reimbursement. And when it's not being physically delivered in the country, you end up in this chicken and egg scenario of where do we access it? How do we access it? If it's not in the country, we won't assess it. So therefore we won't go into the country. Flora, do you want to take the next question or wrap us up?
Yeah, I think there was one question regarding whether we think the experiences of gene therapy in other disorders could be useful. What we are trying to do with Wolfgang and Declan in terms of EARNS, the European group on the rare disorder, and especially on the hematological area, we are trying to collect the data on the benign hematology, especially for example, thalassemia or other type of disorder where there was used the same strategy of AAV. And this is also very important for us to understand how to manage regarding the eventual side effect, toxicity, and collection of the data. And now I want to go back to ask some question by myself from the speakers. I think right now there is a lot of discussion on how really practically we have to do the hub and spoke, because every physician who was in touch with his or her own patients wants to take care of the patient, and this is normal and natural. But we have also to think about the safety, which is very important issue. However, the rate is low, but still we have seen that the liver enzyme could be modified. We could have the problem of the high expression. We could also think about if something goes wrong, we have to be careful about managing and proceeding. So I think, do you think as a physician that every physician in hemophilia are really understanding how could be the management of the patients with the gene therapy if there is any complication over there? Because I think that's the way people should think. I want to make sure my patient is safe, and I want this person to be in a center. If something goes wrong, I can take care of him. The worst thing would be to have the patient who does not know really to who refer. So do you think we need, guys, to work on that aspect and give this responsibility to the centers who want to carry the management of the patients? I want to hear your feeling about that. Who wants to start? Wolfgang, again, you are the first centers.
Yes, this is a great question, Flora, again, and I I only can share my same impressions that this is the most important thing to have the possibility to detect as early some adverse events and then to know how to manage it. You know that we only have very limited experience with gene therapy, each of us, so has only treated a couple of patients, and luckily in Frankfurt it went very well, but I think in the future, especially after approval, we need not only a registry or data collection, maybe also would be necessary to have a collection of some adverse events that we can collect more experience, and also maybe from this point of view, management plans can be developed for different scenarios. I think the ALT elevation is not the same in every patient, so it can occur very early, it can occur after 6 months, it can also occur after 1 year. And is this really the same underlying reason in each of these ALT elevations? There are some ALT elevations where immune reactions can be detected, and there are some ALT elevation without any immunological or inflammatory reactions. So probably we are confronted with different patterns of ALT elevation, and probably these different patterns of ALT elevation need different management ways. And for this, I think in the future we need a much more data collection, and hopefully this will answer the question we have so far.
Thank you, Declan. Or Yesim?
Well, I fully agree with your view. The safety of the patients is the key question on that, and we all have very few experience. And just to take the purpose of the Professor Miesbach, the ALT variability, when I'm talking with our hepatologists, they say, guys, this is really physiologically very variable. So as we will define what is safe and what is not safe, So all ALT variations will not be the same. And we need really the baseline ALT levels of patients, not only once, but maybe several times, to see the individual variations of the same individual, and then decide for this person what is safe, what is not. And we definitely need to pool our data to learn together from the experience of others, and not only our own centers or our country.
Thank you. Declan, what do you think is a feeling of the patients regarding that aspect? I mean, how you think we can work in the future?
Yeah, so this is a really interesting discussion at the moment, and I see it in very rare diseases where patients do travel, and it's acceptable based on the risks associated with getting the therapy. So it's not as simple as a IV infusion. It's either an infusion directly into the central nervous system. There's no other option. You want a real expertise around lentivirus, whatever it may be. They seem to be the drivers as to why there needs to be a small number of centers, and therefore that rationale is okay for patients, for some patients to travel where possible. In other cases, in our situation, it's a simple IV infusion, so it actually is quite easy to give. And I think on a national level, there should be an ability to have access to therapies with, not too far from you. So many hub centers across Europe, and the problem with international hubs is that we're going to face the legislation of trying to get reimbursement across Europe, and that slows down access. It slows down problems. So I can see a benefit in terms of expertise. I can see a benefit in terms of getting faster access, but the difficulty becomes much more in the legislation, and patients, as long as they have the same sort of situation as what Peter highlighted, where you can go maybe for 2 or 3 days and then you're back and you're fully monitored, I think that's acceptable as well. So it'll depend on the patient, it'll depend on the country, and will very, very much depend on the reimbursement. And I'm going way over, so I'm going to take the lead for the end. Thank you to Professor Dargaud, Professor Miesbach, and Professor Kampmann for giving us really fascinating talks on hub and spoke and how it might be practically put into place within your country or across countries. And I want to leave it with the thoughts from Peter's slide on what's important to the patient. The spoke, or my home center, is my relationship, is my life, is the way we've gone. But I really want that delivery of gene therapy to be smooth, professional, and experienced. And we have to find ways to make those centers work together and not have this feeling that it's a third tier or a second tier of centers, and that we're all really collaborating in the same way as Yesim talked about in her opening slide. So with that, I will finish up the webinar, and thank you very much for attending, and hopefully we'll see you again at the next webinar for EAHAD and EHC in the coming months. Thank you very much.
Thank you. Bye.
Bye now.
Thank you so much. Bye-bye.
Bye.